Hello everyone, this is Dr. Madhushri Chatterjee and welcome to XII Biology classes. Today's lecture is on reproduction in organisms. This is the first chapter of class 12 biology. So without wasting time, let's get started. Each and every organism can live only for a certain period of time. Now, the period from their birth to the natural death of an organism represents its lifespan. Plants generally have greater lifespan as compared to animals. Say, for example, sequoia or redwood tree. It can live for 3,000 to 4,000 years. This is because the plants generally continue to grow by their tips. Here in this slide, the approximate lifespan of some plants and animals are given. Isn't it interesting to note their wide variation? Have you noticed the lifespan of crow is 15 years, whereas that of parrot is 140 years. That means lifespan of organisms should not be correlated with their sizes, right? Because crows and parrots are not very different in terms of their size, but their lifespan is showing great difference. Here I have mentioned few extra points for NEET exam. Lifespan usually includes 4 stages. Stage number 1, juvenility. During this stage, the organisms develop capacity to reproduce. Then comes stage number 2, that is maturity. Reproduction begins during this stage. Third one is aging and senescence. Now, aging is the progressive deterioration in the body of the organism, whereas the terminal irreversible stage of aging is called senescence. So I think now it is clear to you the difference between aging and senescence. Then the last stage is death. Senescence finally leads to death. We should remember something that is very important, and it is the changeover from one stage to another is determined by genetic as well as environmental factors. Then point number 2, I have mentioned that there is no individual which is immortal except some single-celled organisms like amoeba. Now, can anyone tell me why amoeba is immortal? If you know the answer, please write in the comment section below this video. Then sequoia or redwood tree lives for 3,000 to 4,000 years. This already I have discussed. And the last point is, oldest plant is Laria tridentata. It's 11,300 years old and it is found in California, USA. See, I have mentioned that no individual is immortal. Then how does the vast number of plants and animal species have existed on Earth for several thousand years? It means there must be some process in living organisms that ensure their continuity. Yes, I am talking about reproduction, and this chapter also talks about reproduction. Reproduction is nothing but a biological process in which an organism gives rise to young ones, that is offspring, similar to itself. Now the offspring will grow, it will mature, and in turn produce new offspring. That means reproduction enables continuity of the species generation after generation. Now there are two main types of reproduction, asexual and sexual. For your easy understanding, I have made 2 columns here showing the differences between these 2 modes of reproduction. Asexual reproduction, a single parent is involved. That is, it is uniparental. In case of sexual reproduction, 2 parents of opposite sex, that is a male and a female, are involved. And it is known as amphimixis. Point number 2, in case of asexual reproduction, no formation and fusion of gametes take place. Whereas in case of sexual reproduction, formation and fusion of gametes take place. Point number 3, asexual reproduction, mitotic division is involved, whereas in case of sexual reproduction, it involves meiosis. Last point, individuals in case of asexual reproduction are morphologically and genetically identical, and these are known as clones. And remember, members of each clone is called ramete. Whereas in case of sexual reproduction, individuals show variation. They are not identical to each other. Now, asexual reproduction occurs normally in single-celled organisms like Monera and Protista. Okay, can anyone tell me what is the basic difference between Monera and Protista? If you know the answer, write in the comment section below. Asexual reproduction is absent in all the vertebrates. Now, there are different modes of asexual reproduction. First method of asexual reproduction is fission. It is the division of the parent body into two or more daughter individuals identical to the parent. Now, fission is of three types: binary fission, multiple fission, and plasmotomy. So let us begin with binary fission. Binary fission is again divided into four types. First one is simple binary fission. That means it can occur through any plane, as for example amoeba. Second one is longitudinal binary fission. It is found in Euglena. The third one, transverse binary fission, is found in Paramecium, Planaria, diatoms, and bacteria. And the fourth one, that is oblique binary fission, where the plane of division is oblique, it found in Seracium, which is a dinoflagellate. Then the next one, that is multiple fission. In this process, the parent body divides into many daughter individuals, that is multiple individuals. It is found in amoeba under unfavorable conditions. And the third one is plasmotomy. What happens in plasmotomy? It is the division of a multinucleate parent into an multinucleate individuals without division of nuclei. But nuclear division occurs later on to maintain normal number of the nuclei. Now it is found in Opelina and Pelomixa. Remember, both are protozoans. Then the second method of asexual reproduction is budding. Budding in yeast is known as torulation. Why? Because this budding stage resembles with a genus Torula. Budding is also found in animals. A daughter organism is formed from a single projection arising from the parent body. Now, in animals, budding is of two types. One is exogenous or external budding, and as the name suggests, here the bud grows externally on the surface of the body. It is found in Hydra, Cycon, in certain annelids like Psyllis, tunicates like Salpa and yeast. And the second one is endogenous or internal budding, which is also known as gemule formation. Here also, as the name suggests, buds are formed within the parent's body. That is why it's called internal. It is found in freshwater sponges like Spongilla. Now see, I have mentioned a very important point for NEET exam. Gemules, that is internal budding, consist of small group of cells which are known as archaeocytes, and they are enclosed by a protective coat. Now, during favorable condition, the mass of archaeocytes come out through micropyle and later on forms new colony. Now, remember, archaeocytes are important for your NEET exam. Then the third method of asexual reproduction is fragmentation. Here, the body of the parent may break into some pieces. That is, the body gets fragmented and each piece or fragment develops into a new animal. It is found in certain flatworm, sponges, coelenterate, algae like Spirogyra, fungi like Rhizopus, bryophytes like Riccia, Markentia, pteridophyte like Selaginella, etc. Remember, you need to memorize the examples very well, okay? Then the next mode of asexual reproduction is gimme. Gimme are specialized structures which are green multicellular asexual buds that develop in small receptacles called gamma cup. Now singular term is gamma and the plural is gimme. Now the gimme are separated from the parent body and germinate to form new individual. And yes, it is found in Markentia. Then the fifth mode of asexual reproduction is regeneration. Now, regeneration is the formation of whole body of an organism. It can be done from a small fragment and then it is known as morphollaxis, and it can be done with the replacement of the lost part. Then it is known as epimorphosis. Regeneration occurs in amoeba, sponge, hydra, and planaria, etc. Here again, I have mentioned an important point for NEET, that is regeneration was first discovered in hydra by Abraham Tremblay. Okay. Then the next mode of asexual reproduction is spore formation or sporulation. Now, spores are minute, single-celled, thin-walled propagules which germinate to produce new individuals. Some spores are briefly described here. Let's begin with zoospores. Now, zoospores are special kind of motile spores. Now, since they are motile, they should possess flagella and they are produced inside sporangia. Zoospores are generally naked, that is without cell wall. Reproduction through zoospores are found in some lower fungi like Phycomycetes and algae. Now here I have a question for my viewers. Why Phycomycetes is known as lower fungi? If you know the answer, please write in the comment section below this video. Then the next type of spore is conidia. Conidia are formed in penicillium. Conidia are non-motile spores. Then the third one is chlamydospore. It is thick-walled. It stores reserve food material capable of withstanding long unfavorable conditions. Example is Candida albicans. The last one is sporangiospore. It is non-motile spore and it is found in Rhizopus. Then the next mode of asexual reproduction is vegetative propagation. It is the formation of new plant from vegetative units called vegetative propagules. Remember, this method produces a large number of population of clones in a shortest time. Now, vegetative propagation can be achieved by two ways. One is natural, another one is artificial. Now, let us start with the natural process. First is by root. Both tap roots and adventitious roots take part in vegetative propagation. As for example, dalbergia or shisham, then guava, poplar, sweet potato, tapioca, dahlia, asparagus, etc. The second one is underground stem, like tuber of potato, bulbs of garlic, onion, etc. Corms of Amorphophallus or zaminkand, colocasia, etc. Rhizomes of banana, ginger, turmeric, and suckers like mint, chrysanthemum. Here, I would like to give you a suggestion. That is, you can have a very quick revision of your 11th standard biology textbook where you studied a chapter named Morphology of Flowering Plants. In that particular chapter, this kind of modifications like tuber, bulb, corms, rhizomes are mentioned. So my suggestion would be, please go through that chapter once to get a clear understanding of this part. Then is subaerial stem. These are three types: runner, found in lawn grass, then stolon, as found in strawberry vallisneria, then offset, as found in ecornia or water hyacinth, pistia or water lettuce. Now here, as you can find, I have given a very important point for NEET exam. That is terror of Bengal. Yes, water hyacinth is called terror of Bengal. Why? Because it is the most invading weed found in growing standing water. And it takes all the oxygen from the water, which causes, of course, the death of the fishes. Now, this plant was introduced to India for its beautiful flower and shape of leaves, but it can propagate vegetatively at such a fast rate that it can spread all over the body in a very, very short period of time. Then by leaves is found in begonia, bryophyllum, kalanchoe, streptocarpus, adiantum, etc. Here again, one important point for NEET has been mentioned, that adiantum caudatum is called walking fern. Why? Because its leaf tips form new plants when they come in contact with soil. Then it can be achieved by bulbil also. Now what is bulbil? It is multicellular fleshy bud and found in oxalis, agave, dioscorea, lily, etc. And the last one of the vegetative propagation is through turion. Turions are swollen bud containing much stored food, as for example in a number of water plants like Potamogeton, Utricularia, etc. So here I have given diagram of few tuber like then offset, then runner, etc. I think you will find these diagrams in your textbook as well. Now the second mode of vegetative propagation, that is artificial method. Now these are man-made methods of vegetative propagation, and this is also known as horticultural propagation. Now let us begin with the first process, that is cutting. Now cuttings are cut pieces of root, stem and leaves which are planted in nurseries. And for this, root promoting substances are used like IBA, say indole butyric acid, then NAA, naphthalene acetic acid. Okay. Now cutting, first let us begin with root cutting. Root cutting is used in the propagation of lemon, orange, blackberry, etc. Then stem cutting, it's a common method of plant propagation and found in rose, sugarcane, duranta, bougainvillea, etc. And leaf cutting is found in sansevieria, begonia, bryophyllum, etc. Then the next method is grafting. It is very important because it is the most common method of vegetative propagation. Now grafting happens between two plants of closely related varieties. Now plant from which root system is taken is known as stalk, and the plant from which shoot system is taken is called a scion, and this forms a composite plant. It is found in mango, apple, pear, citrus, etc. Now you should remember something, that is stalk is always older than scion, and scion is generally taken from the plant having superior character. For example, a scion of Daseri mango is grafted on the stalk of Desi mango. Similarly, scion of high quality roses are grafted on wild rose root stalk. Okay. Now during grafting, stalk and scion both are united by callus, which is produced by cambium. That is the reason why grafting is successful in dicot and unsuccessful in monocot, as monocots have closed vascular bundle, that is lack of cambium. Please remember, this is very important for NEET exam. Okay. Then the next method of artificial vegetative propagation is layering, that is found in jasmine and strawberry. And the last one is propagation by tissue culture. Here I am not going into the detail of this tissue culture method because completely dedicated chapter is included in your syllabus, where you are going to study about the whole process of tissue culture. Okay, so at that time, I will discuss about tissue culture method. Now let us discuss few merits of vegetative propagation. First one, it is the only method of multiplication in seedless plants like sugarcane, banana, seedless grape, etc. Second one, it's a rapid multiplication process. Third one is it shows genetic uniformity. And the fourth one, plant tissue culture is used for producing disease-free plants and transgenic plants as well. There are some demerits as well, like it has no variation, it causes overcrowding, and vegetative propagules are prone to viral, bacterial, and fungal diseases. So here I have given a list of few questions. You know what you have to do. Yes, you have to write down the answers to this question. So I would suggest just pause this video and take down all the questions in your notebook and start writing the answers. Because see, this class is regular batch, okay? So in addition to NEET, here the parts that are important for your board exams will also be taught. So these questions are quite important for your board exam. And now I have given 3 critical thinking questions, and don't worry, the answers are already discussed in this video itself. Okay. Question number 1, why is the organism undergoing binary fission immortal? Question number 2, why is phycomycetes called lower fungi? And question number 3, why is grafting unsuccessful in monocots? So you can send your answers at my email ID that is given here, madhushri_chatterjee@hotmail.com, and I will definitely check your answers. So if you find this video helpful, like this video and subscribe this channel for many more useful videos. And the next class is going to be on the part 2 of this chapter. Okay, thank you so much.